Hey everyone, welcome to Science in Perspective. So there is a story circulating right now. It's repeated in front of world leaders. It's backed by hundreds of millions of dollars in funding, and it's carried by some of the most respected names in science. And it basically goes like this. Aging has a simple explanation, and we are close to reversing it. So today we're going to follow that story. It's not so much to tear it down, but to use it as a lens, because inside it is one of the most important errors you can make when reasoning about any complex system. Super important to understand that. So we're going to look at what aging actually is, what the experiments actually show, and why the gap between those two things matters far beyond just longevity research. So I'm going to leave you with something towards the end which you can actually use. It'll be like a named reusable concept that applies anywhere or anytime someone claims to have found the single lever that controls a complex system. And this appears all the time. I want to make sure you're on the lookout for it. You know how to recognize it. So the core insight here in this episode is that aging is not a file you can restore. It's a process being continuously enacted across many interacting systems. And once you see that, the phrase age reversal or something like it just stops making sense. Not because the science in some isolated sense is bad, but because the concept itself is actually incoherent. So before we go in, let's just kind of run through the core argument of this episode so you know where I'm about to take you and make sure you're interested. And then we'll kind of dig into each piece and see where that takes us. So as humans, right, we're wired to believe complex systems have a master switch. This is just how people think because we're not used to really understanding complex systems in an analytical way. We're very much used to understanding complex systems natively, intuitively, because that's the environments we grew up in. That's what we evolved for us, what our ancestors were surviving in. But when you kind of take the Enlightenment approach and the scientific method, if you will, all of a sudden we're always looking for root causes. Doesn't apply super well to complex systems. So the simpler a story sounds, the harder it is to question. Now, aging specifically is not one thing, right? It's a distribution of failures across many interacting systems, and it's measured through proxies that are not the system itself. The most prominent aging researcher in the world, which we're going to highlight in a bit, is reasoning from a kind of CD and scratches metaphor that the underlying biology cannot even support. I'll let you know what that is in a bit. The actual experiment behind age reversal is like one tissue, one injection, one isolated subsystem, but it ends up getting mistaken for command of the whole organism. That's the way it gets spoken about. Sometimes within scientific circles, and definitely to laymen, when these things get popularized. But again, in this episode, it's going to be the leading researcher making some of these comments and these analogies, which really give the wrong impression of how well the research is going. Stored things can be restored, right? But enacted processes cannot. I'll define enacted in a bit. Aging lives at the enacted layer. It's written moment to moment by everything else in the body. Basically, the identity of a thing, when I say enacted, is something that only takes on that identity if it keeps moving. It's a very dynamic thing. It's not something you can point at. It's not something you can kind of polish and restore. It's just not how complex systems work. So the error, I'm going to give it a name. I'm going to call it the single layer reset fallacy, so we can kind of anchor onto something. Once you have that name, you can see it in economics, you can see it in AI, you can see it in medicine, and I'd argue you can probably even see it in your own life in certain situations. So we've got this problem. Like I said, we're wired to believe that complex systems have a master switch. Our brains are pattern completion engines, right? And the simplest pattern is always going to win first. Now, I was kind of blaming this on more of the Enlightenment scientific overanalysis that we tend to do, which, again, works really well in reductionist systems, not so well in complex systems. But even from an evolutionary standpoint, right, we like narratives, we like linearity, we like agency, we point to the thing that we think is the cause. But we know this can get us into trouble if we're misplacing that concreteness, so to speak, right? Okay, so to anchor this kind of whole episode, there is a Harvard geneticist, and he told the world recently, actually told world leaders, that aging has a simple explanation and is reversible. This guy is David Sinclair. You may have heard of him before. He's a professor of genetics at Harvard Medical School. He's the co-director of the Paul F. Glenn Center for Biology of Aging Research and co-founder of Life Biosciences. That's a Boston startup that in January 2026 actually received FDA approval for the first human trial of an epigenetic reprogramming therapy. We'll get into some definitions in a bit. He coined what he calls the information theory of aging, the idea basically that aging is fundamentally a loss of epigenetic information. It's not so much an accumulation of genetic damage as many of us and many scientists might think. He's the author of a book called Lifespan, Why We Age and Why We Don't Have to. That's by Atria Books in 2019. And that's kind of really where he brought his ideas to the mass audience. So this is not a fringe figure, right? He's arguably the most prominent longevity researcher alive right now, which is exactly why his framing matters and why examining it carefully, I think, is worth our time. Okay. So again, 2019, you got this book coming out. It's called Lifespan. You can take a quote right out of there. To become young again, we just need to find some polish to remove the scratches. Okay. So, again, this is kind of where this was first kind of introduced to the general public, I guess you could say. And the book's subtitle is itself a claim, right? The argument inside is that aging is a disease. It's almost like it's an engineering problem that can be solved, right? Or at least a very locatable, specific root cause disease that we can approach. It has a single upstream cause, you could say, which Sinclair would say is loss of epigenetic information. And therefore, it should be treatable and reversible, right? So again, that direct quote, to become young again, we just need to find some polish to remove the scratches. Now, you can say, yeah, okay, that's just it's a metaphor. It's a bit of an analogy there. But, I get what he's saying, right? I mean, there's information involved in biological systems, and you're just doing something with the information. If you can kind of reset it, like a compact disc that has scratches on it, if you can do something to get those scratches out of there, can't you recover the information, right? It doesn't sound crazy if you don't know anything else about the systems, right? Okay, let's continue with some of the stuff that David has said publicly before we get into and kind of pick apart why the analogy is wrong and a better way to think about this. There was a TEDx Boston 2022 talk. This was titled, Is Aging Reversible? A Scientific Look? Again, by Sinclair. The title of the talk is the claim, right? It's not, might aging be reversible or could we slow aging? The framing is settled and the talk is positioned as a scientific confirmation. And you can actually go access that full transcript publicly. It's available at... I'll put the link in the description, the singjupost.com. But again, so this is the moment, right, that you kind of move from the book claim, just kind of generally introducing it, into a mainstream public platform with the TED brand attached to it. Okay, that's why this stuff is important. It's not just some isolated researcher doing their thing and maybe making some loose analogies, right? This is the stuff that goes out to the general public and also to the other, the rest of the scientific community, which wouldn't be involved in longevity research and might think that certain things are real that are not, right? It's important. It's important to point it out. Again, it's not attacking necessarily the science itself, necessarily. Like I'm sure Sinclair is doing good science. I'm sure there's good stuff. There's good discoveries being made. It's important information. But you got to be careful about the way you frame it because it completely can misrepresent how something like aging or life in general or the human body works. It's important to get that right. So let's do some other examples. 2023, there was a post on X where Sinclair posted that age reversal and vision restoration had worked in primates and that humans were next. All right, now, this is the first time the claim kind of jumps from animal models to a direct implication for human trials. And again, it was posted publicly to his large following without the caveats that would accompany something like a peer-reviewed paper. Right? Now, again, you can say, yeah, he's just excited, right? He's an optimist and he wants to push this forward. Fair, but you still got to be careful about not suggesting that things are possible that are not, or definitely currently not. Even not ever possible. And I'll tell you what I mean by that. Again, not that the science itself is wrong, but the way it's getting extended is those things have a single lever you can pull, or though one good example here means it's going to be something that works writ large. It's just not true. So, Now, it's not just me, right? There was pushback from within the longevity field itself, and that began to escalate seriously, arguably since this X post. That's kind of when it first got triggered, right? So it wasn't just buried within a book. It wasn't just some niche kind of thing. Actual other longevity researchers were like, okay, hold on. Let's not say things that are pseudoscientific. Let's make sure we keep this real. There was an Animal Bioscience press release in February of 2024, and there was actually a resignation that followed. So this is another company that Sinclair co-founded called Animal Bioscience. And it quoted him in a press release claiming that a supplement had reversed aging in dogs. You might remember this. And then again, again, fellow longevity researchers kind of pounced on this, not outside critics, but people from within his own field, and they basically responded with a wave of formal rebukes, right? So they're not happy, which you shouldn't be, right? Like, I mean, if somebody's working in your field, and even if they're one of the most well-known and established and celebrated researchers, that's great, but careful, don't start saying things that are just simply not true, that makes the rest of the field bad, look bad, it's not going to be taken seriously, right? March 2024, Sinclair actually resigned as president of the Academy for Health and Lifespan Research under that pressure, because he said those words, and because people in the longevity community were not happy. And he also pledged to be more scientifically accurate in future public statements. So he's aware of this, right? It's not like he's completely pushing back and saying, no, it's true. You got to be, so this is just more about being careful how you speak, right? And it is critical. It's important because the general public doesn't know that. They hear scientists speak and they think, oh, I guess that's how science works. This is great. Not to mention who you vote into power and what gets funding, what policies get put into place. This all comes down to not just fundamental research, but kind of the popularization and the way that science gets spoken about. You do have to be careful. So this is kind of the key piece of backstory for this episode here, right? The pattern was flagged. There were consequences that followed. And then, less than 2 years later, the same framing reappeared on an even bigger stage. And so we can kind of think about what that escalation looked like. What it was. So another post on X, January 27, 2026, there was this exchange with Elon Musk. Elon Musk told a Davos interviewer that aging is very solvable, and that the answer will probably turn out to be obvious. And then Sinclair replied publicly on X, aging has a relatively simple explanation and is apparently reversible. Clinical trials begin shortly. Okay, Elon Musk, right? Entrepreneur, optimist, not a scientist, right? In any full-time sense of that word. Okay, he's going to use he's going to be pushing optimistic type wording onto the general public, and we kind of get that. But then when the scientist, one of the leading scientists in that area, has a chance to maybe temper the wording a bit, he just supports it and then just pushes it. Okay. And anyway, so these are examples, right? Posted to an audience, right as the FDA trial was being announced. It's not like it's a journalist's paraphrase. It's not taken out of context. It is the claim in its clearest form from the scientists doing the actual work. Let's do one more. There's a World Government Summit in Dubai, February 5th, 2026, super recent. There's a session titled The Science of Living Longer and Better. 9 days after that X post I just mentioned, Sinclair stood in front of assembled heads of state and senior government officials and said, "We are about to test for the first time in history whether we can reverse aging and cure diseases." And he also predicted that within 10 to 20 years, modern healthcare systems will look outdated, and kind of described this moment as comparable in significance to the discovery of clean water and vaccines. Okay, so this is where the claim reached its largest institutional audience. It's not a podcast, it's not a tweet. It's a summit attended by government leaders from around the world, and that is the scale of reach we are dealing with. Okay, so why does this matter if it's not obvious already? Well, the simpler an explanation sounds, the harder it is to question, right? Think of an analogy. If you pull one weed and believe that the garden is healed, something kind of wrong with that, right? We've got this single cause attribution in science communication that ends up amplifying this kind of cognitive bias that we all have. We all do this in one form or another. We do it in our own lives. It's hard, actually, not to do it in science, especially if you want to start, if you're not careful, right? Because you want to use analogies, you want to use metaphor, you want to use allegory, you want to communicate effectively, right? If I'm going to talk to someone about quantum mechanics and I want laymen to have a reasonable grasp of what I'm talking about, I'm not going to just throw a bunch of equations on the whiteboard and start talking about all the superposition and entanglement and wave function amplification, blah, blah. No, you might have some of that, but I've got to start using analogies. I've got to, what does this mean? What's a good way to think about it? But you have to be careful when you use the analogies that you're not saying something that's actually unscientific, right? We all have the cognitive bias. We need to be aware of it. Really got to be aware about it in scientific communication. And many of you probably aren't doing longevity research, and so maybe you don't know, right? Like are we really that close to curing aging? Is aging really just a disease, right? Or is that the right way to think about it? Science in perspective, right? Let's put this into perspective so at least I can give you a better idea. Again, I'm not saying the research is all wrong, but let's just put a better, more realistic frame around this. Let's de-hype it so you know what's real. So we've got this problem, right? The word aging hides extraordinary complexity behind a single label. Aging is not one thing. It is a distribution of failures across many interacting systems. Okay, so when researchers study aging seriously, they do not find one thing going wrong. They find a cascade of failures that spread across systems that interact with each other. So you have things like epigenetic drift. Okay, you've got molecular tags that tell your genes what to do. They can become noisier over time, is a way to think about it. So it's like a recipe that slowly accumulates smudges until the cook starts guessing, right, of what he's even cooking. Hopefully that analogy is not too stretched. Protein misfolding is another thing. Like, these are things we know that seem to be associated with aging. We've got tiny machines inside your cells that build, they repair, and they run everything. And they start making errors that they cannot correct. So you get this misfolding of proteins, right? You got mitochondrial dysfunction. Think of it as like little power stations inside your cells. They become less efficient and they start leaking damaging byproducts. And you got immune dysregulation, right? The system that cleans out damaged cells and defends against threats. Those can start misfiring, attacking things it should actually leave alone and ignoring things that it should clear. And I just used a bunch of analogies there. Analogies are great. They're critical. They're very, I would argue, they're, as Hofstadter often argues, they're kind of to paraphrase, the fabric of cognition. I don't think you can have a thought that's not an analogy. You're always kind of making those associations. But you still got to make sure you're not saying something unscientific. You got to make sure you're being realistic about it. And as a scientist, you have to be responsible for that. So the critical point is that these are not 4 separate problems that I just listed. They're 4 coupled processes. It's the coupling that is very much the hallmark of complex systems, meaning things are interacting. If you change one, let's say when one thing degrades, it can actually accelerate the other. So you can't just go to one lever, pull it, and think, oh, we isolated this thing. That's not how it works. And when I say complex systems, I literally mean reality, because everything in reality, in real natural systems outside of the lab, is highly, highly codependent. They don't act as independent pieces, right? So these systems don't fail separately. They co-degrade, you could say, and they can accelerate the others, each of them. Okay? Let's get to some basic definitions before I go any further, because I've said things like epigenetic and some biology terms in there, mitochondrial, yada, yada. Let's make sure we understand what some of these terms mean. You probably know already, maybe, but just in case. DNA, right, deoxyribonucleic acid. This is the type of molecule the body's instructions are written in. It's using a 4-letter chemical alphabet, ATGC. Most of us know what that is, but sometimes this kind of gets interchanged with the word chromosome. It's not the exact same thing. One DNA molecule packaged with proteins into a compact, organized structure that fits inside the cell is a chromosome, right? So humans have 46 of them in every cell. Okay, so you can kind of think of, you take a strand of DNA, you pack a bunch of stuff around it, it's now zigzagged and squished and packaged in a way that needs to fit into the nucleus of the cell where it can actually be used. That's chromosome. We have 46, we have 23 pairs coming from the parents, right? There's also genome, right? That would be the complete sequence of letters carried across all 46 chromosomes taken together. So we know DNA has a sequence, and you can package that up into a chromosome. And then you can take 46 chromosomes, and if you take that whole set and you read the sequence across that whole set, that's what people mean, or scientists mean, when we say genome, right? In humans, it's about 3 billion letters, and it's actually the same in every single cell of your body. Okay, so the word genome is kind of a totality word. It does not refer to a separate object that you can point at. It refers to the complete sequence of letters across all 46 of your chromosomes taken as a whole, right? So 3 billion letters in their specific order, and the same sequences in every cell of your body. Your liver cells and your neurons, they actually carry identical genomes. What makes them different cells is which parts of that identical sequence each one is currently reading. So you can read sequences of the genome, which basically gives it its functionality within whatever system it's in, subsystem of your body. So it's distributed across 46 physical molecules, but it's treated as one complete text, right? So it's kind of the way a multi-volume encyclopedia is one work, spread across many books on a shelf. Okay, so that's a genome, that's chromosome, that's DNA. What about gene? Well, that would just be a defined section of that sequence of the genome that codes for something specific the body needs to build or do, right? Maybe there's genes related to the color of eyes or something. It's not going to be the whole genome, but there'll be a section of it that specifically codes, a section of the sequence, right? Okay, so every single cell carries the same genome, but different cells use different parts of it. This is actually going to be really important when we get into the enacting part and understanding epigenetics. So it's not just all kind of bio 101. It's important that you understand this. So every cell carries the same genome, but different cells use different parts of it. So what makes a liver cell is which genes it is reading right now. Genome's the same, which genes it's reading. Epigenetics, which gets into kind of the language that Sinclair is using when he talks about aging, right? Epigenetics, think of it as the control layer that decides which genes a cell is using at any moment without changing the underlying sequence, okay? So mechanically, how does this work? We got small chemical tags placed on the DNA and on the proteins it wraps around, written and erased by enzymes that respond to signals from the cell and its environment. So if you're getting lost, don't worry. The take-home message there is that this is a dynamic thing. What makes the cell do what it does, what allows the sequences to encode for what they encode for, is not some static thing that sits there, okay? It is a dynamic, always on, always moving thing that gives it that ability, it gives it that functionality. Here's an analogy, okay? So maybe a kitchen and a recipe book. Every kitchen owns the same complete book, but what is actually cooking right now depends on what is open on the counter, what is simmering, and what the customers walked in asking for, okay? So, kitchen, recipe book, every kitchen's kind of got the same thing, but what's actually cooking right now depends on like what's on the counter, what's simmering, what's in the pot, what the customer's asking for, who's paying for what. That's a dynamic thing. It's always moving. That's where the definition actually comes from. So this layer is dynamic. It's not stored. The tags are continuously rewritten every moment in response to what is happening in the body right now. So this is what we mean by enacted, okay? It's a process whose identity exists only in its ongoing operation. It is not saved anywhere. And if the operation stops, the thing does not pause. It ends. So the cleanest way to understand enacted is to think of things in the world that exist only because something is currently happening. And notice that they have no saved version anywhere. A flame is not a thing sitting on a candle. It is combustion. In progress. Right now. So the reason it looks the same from one second to the next is that the process is pretty steady. It's not that the flame is stored. If you blow it out, and you do not get a paused flame, then you can later unpause. You can't blow it out and then, get a paused flame and then later come in and unpause it, right? We get that. You get nothing because the flame was never an object. It was a performance, right? What's another example? I don't know. A whirlpool in a river is kind of the same thing, right? So was a conversation. So was your heartbeat, your attention, your sense of who you are right now. None of these are saved files. They are all enacted. They exist only as their ongoing operation. So now hold that picture against the layer Sinclair is claiming to reset. This is why I'm giving you this kind of background, right? The epigenome is not a file inscribed in your cells at birth that has slowly accumulated scratches. That's literally not what the system is. It is the moment-to-moment output of enzymes deciding right now which genes to expose for reading, based on signals coming from a body that itself has been drifting for decades. The youthful epigenome was never stored anywhere. It was being performed every second by an orchestra that had since drifted into a different style of playing. So there's no recording to revert to because the original was always a performance, not a recording, right? So a flame, a whirlpool, a conversation, they all exist only as its current performance. If you cut the throughput and you do not, if you do that, you're not going to get a paused version. You just get nothing. Okay. It's true. That's not just a metaphor, right? Like if you look at those systems, that is 100% correct, right? So stored, let's do the contrast now. What does it mean if something's stored? Well, information inscribed on a passive medium that persists whether or not anything is happening to it, right? A file on a disk, a sequence written down, a CD on the shelf, right? So this distinction is super, super important. Sinclair's framework needs aging to be stored because only stored things can be restored, right? Restored from a saved version, right? What an epigenetic clock actually is, though, a correlated proxy, not the aging process itself. So an epigenetic clock is a statistical tool, essentially. It looks at chemical marks on your DNA called methylation patterns and produces a number that correlates with how old your cells appear to be. It is genuinely useful as a research instrument, but here is where it is not, or what it is not, rather. It is not aging itself, right? It is a shadow of aging. It's a correlated signal. When researchers report they have reversed someone's biological age by 10 years, and you hear this a lot, kind of just casually, maybe on X or other platforms, but unfortunately sometimes by scientists as well, what they actually mean is that the clock reading went down, right? The underlying processes that drive the clock, the protein errors, the immune drift, the mitochondrial wear, they were not even directly measured. They were inferred through the proxy. So this matters enormously, right? This is not a simple thing. A proxy can move without the underlying system moving. And an underlying system can continue moving while the proxy appears stable. The clock is a useful map, but as every complexity researcher knows, the map is not the territory, if you've heard that before. Okay, so this proxy distinction is super critical. Measuring a shadow is not measuring the object that is casting it. Measuring a shadow is not measuring the object that is casting it. A credit score tracks financial health, but it is not your financial health. It's one little piece of it, which is codependent on a bunch of other things happening in your life related to finance. And maybe not even related to finance. Okay, so the most prominent aging researcher in the world is reasoning from a broken metaphor. I think that's a problem. Sure, he's a great scientist, but this as soon as you start speaking to the public, right, this is a problem. Sinclair's claim that aging has a relatively simple explanation and is apparently reversible, that is seductive, right? The CD, compact disc metaphor, sounds exactly right on first contact. I'm assuming everyone knows what a CD is. I guess I'm dating myself a bit, but the compact disc metaphor from the book Ultimately Lifespan, 2019 book, right? It's like aging is scratches on a disc. If you polish it, you'll restore the original music, right? And that's how Sinclair is describing aging. Now, hopefully you understand with all this enacting kind of description I'm giving you, and the dynamics and how complex systems actually work, you see the problem with that, right? And again, it's not just the book. He's repeating it in nearly every public appearance since he's written that book, right? Aging is just scratches on a compact disc, and it's like it makes the music skip, right? Is there a backup copy of the epigenome? In other words, can you polish that CD and get back the original music of your youth? There's nothing to get back. It doesn't make sense, right? From that book directly, quote unquote, to become young again, we said this, right? We just need to find some polish to remove the scratches. It is a beautiful metaphor, and this is so I got to be careful with metaphors and analogies. They're quite poetic, right? And often very powerful and true. But in this case, it's also the source of the error, right? The reason it is seductive is that it captures something real. Epigenetic marks do become corrupted over time, correct? And there is a sense in which information is being lost, but the metaphor carries a hidden assumption, right? That there is one layer of information, that it can be separated from the medium, and that the medium can be restored without changing the information. None of those assumptions hold for a living organism. None of them. This is why this compact disc analogy fails, right? Compact disc has one layer. A living system has interacting layers that continuously rewrite each other. If you put that CD in, how does it work? It stores information in a single physical layer. There's pits and lands that are etched onto the surface, right? It's like reading a record, essentially, but you're doing it with lasers. There's no feedback between the encoding and the playback, right? The disc does not change while you listen to it. A living cell is literally the opposite of this. It has no separable layers. Its epigenome shapes which proteins actually get made. Those proteins reshape the epigenome. Okay, so the immune system reads signals from both and changes how it responds, which feeds back into metabolic state, which changes which genes are even accessible in the first place, and that changes protein production again. You understand the feedback loops, right? It's very nonlinear. Every layer is continuously rewriting every other layer. There is no stable substrate underneath, waiting to be uncorrupted. The system is the interaction. So this is the core reason that this kind of factory reset metaphor fails. You cannot reset a process. You can only perturb it. Okay? But this fatal assumption here, right? There is no original copy to revert to. So the most powerful assumption inside the reversal narrative, reversing aging narrative, is also the most invisible one, that there is an original state to revert to. In a backup system, this makes sense. The original file existed, it was stored, it can be restored. The present state is a deviation from an archived baseline, but a living organism was never archived. There is no snapshot of your cells at age 25 sitting in a vault somewhere. What existed at 25 was itself the product of a developmental process. So what exists now, it's just the product of 50 more years of that same process, however old you are, right? The system has moved continuously, and every state was legitimate. There is no original copy. There is only the current state of the ongoing process. That's it. And this is the whole point of enacted, right? You stop moving, just like the flame on the candle. You stop moving, it has no existence anymore. So the question of which direction, you can nudge it and which lever you can pull. Okay. So the actual trial is a tiny fraction of what age reversal implies, whatever little experiment, whatever trial they're doing. What the experiment actually is like one tissue, one injection, 1 8-week window in a privileged subsystem, right? So the first human trial, the one generating headlines about age reversal, what did it actually involve? What was it? Well, there was a target tissue, retinal ganglion cells in the eye. There was a delivery method, direct injection into the eye. There was a mechanism, partial activation of the Yamanaka factors for 8 weeks. I'm not going to go into all the details, but it was controlled via antibiotic, doxycycline, I believe it was. And there was a primary goal: if you restore some visual function in patients with glaucoma or optic nerve damage, and you can show the change. The eye was chosen deliberately because it is isolated, it's accessible, and it's a contained system, right? This is the thing with experimentation. People kind of take it as this default stance that experimentation is always the real world validation of a theory. Well, sometimes, but experimentation is extremely contrived in many ways, right? It's isolated. It's more accessible than it would be otherwise. It's contained. It's actually quite unnatural. As the researchers themselves noted, they even noted it was an easy choice to target because they knew how to administer the therapy's safety there. That's not a criticism. This is good science. You start small, you prove a mechanism, and you expand it carefully. The problem is not the experiment so much. The problem is calling it age reversal. Okay? We know why the eye was chosen. It was isolated, it was accessible, it was deliberately bounded. And what Sinclair's own field said was that, the jump that aging is a problem that can be wound backward is just not justified. Okay, you can go look that up. It was buried in the coverage of these studies. Right? Almost always in the final paragraphs, in the studies themselves, are the voices of researchers who were not involved. And they're actually worth reading carefully. Aging, quote unquote, is a complex process with multiple contributing factors, and the effects of OSK treatment were moderate. A small extension of lifespan in one study and a partial reversal of artificially induced symptoms in another. The jump that now aging is a program that can be wound backward is not justified by the research. That was by molecular geneticist, Jan Vij, at the Albert Einstein College of Medicine. And I'll put these links in the description, but you can go, again, it's not just me, right? Like, there's people in longevity that are pushing back on this. Okay. And so what is he saying? Well, the effects were real, the effects were modest, and the conceptual leap from this marker move to aging is reversible is just not supported, like full stop. So resetting a clock reading is not reversing the process that drove it. If you put a thermometer in ice water and declare that the room is now cold, I think we all get that that doesn't make sense. Right. So imagine a room, it's been slowly heating up for 50 years. You have a thermometer on the wall that reads 98 degrees Fahrenheit. You take the thermometer off the wall, you hold it under cold water, and the reading drops to 72. Did you cool the room? Obviously not. Right? So again, this is not super counterintuitive stuff. We just have to appreciate that this is what complex systems actually are. In this case, you change the measurement. The room is still 98. The process generating the heat, the insulation, the equipment, the sun, they're all still running. Resetting the something like an epigenetic clock in retinal cells, it's the same operation. The reading changes. The processes that drove the reading, the accumulated protein errors, the metabolic drift, the immune state, they're still running everywhere else in the organism. Changing the instrument is not changing the system. Okay? So we can call this error something like proxy collapse. You're mistaking the indicator that you actually see or measure for the system that it's trying to track. Proxy collapse is what happens when a measured indicator gets treated as the system it tracks, and it shows up everywhere. Treating a test score as intelligence, I argue about the IQ all the time. treating GTP growth as human flourishing, right? Treating a stress hormone level as stress itself, treating an epigenetic clock reading as biological age. Proxies are useful. They're used all the time in science. You almost have to use them. The error is not in having them. It is in forgetting that they are proxies. Once you forget, you start optimizing the proxy directly instead of the system it actually tracks, right? And when you do that, you can produce enormous movements in the measurement without touching the thing the measurement was supposed to represent. This is not a new mistake. It's arguably the central mistake of every field that has tried to reduce a complex system to a single number. It's never worked. Never will. There's fundamental reasons for that. So the problems, stored programs and enacted processes are fundamentally different kinds of things. A file can be restored because it was encoded into a static medium at a point in time. Aging is not a file. It is the ongoing result of billions of interactions continuously modifying each other. And that's the distinction that you have to understand, okay? And I think that we get that. I'm not going to kind of belabor that seedy metaphor, but I want you to really understand how aging is an enacted process, right? Even a reset epigenome in one tissue is surrounded by a proteome, immune system, and metabolism that have not changed. So this makes the whole organism reversal conceptually incoherent. It's not even technically hard to understand. It's just it's not technically hard to achieve. It's not like, oh, this is just a really hard problem to solve. No, it's not a problem to solve at all. It doesn't exist. You're not solving a problem. The problem that you're saying exists isn't. It's almost like a straw man in a fallacious argument, right? Think about forest succession or replanting trees. You don't just restore old growth because the soil, fungi, and fauna have all moved into a new state, right? Ecologists have a concept called succession. The way ecosystems change over time as one community of species is replaced by another. A field becomes a meadow, becomes a shrubland, that becomes a forest. Okay, so now imagine someone wants to restore a forest that has been clear-cut back to the old growth, so they replant the trees, and the saplings grow, and the canopies begin to close, but it's not old growth. The soil chemistry has changed, right? The mycorrhizal fungal networks that old trees depended on are gone, right? The seed banks are depleted. The fauna that co-evolved with those old forest trees and what have you, they're absent, right? The new trees are growing into a completely different ecosystem state, one that happens to have trees in it. So you can restore the indicator, which is, let's say, the tree cover, without restoring the actual system itself. The system has moved, and it's in a new state, and that's a product of everything that has happened in between. All right, this is exactly the situation with epigenetic reprogramming in a single tissue. You can restore that indicator, but the organism has still been aging everywhere else, and that's the point. Okay. And there's a bunch of other research you can kind of look into on that, but I think I drove that point across, and I kind of wanted to. I said at the beginning, I'll leave you with something. So because this happens a lot, not just in longevity research, right? We need a reusable tool to kind of catch this error whenever it appears. Call this the single layer reset fallacy, the belief that resetting one informational layer resets the system, right? Think about it like if you reset one informational layer of a complex system, then you're kind of resetting the system as a whole, right? That you think of that fallacy as basically having three parts. So you got the layer isolation, where you treat one measurable subsystem as if it operates independently. It doesn't. I don't care what system you're looking at. If it's complex, it doesn't. Proxy promotion. You treat the indicator for that layer as equivalent to the system state. And you have this kind of control inference where you conclude that movement in the indicator equals command of the whole. You see this error in longevity research. You also see it in economics, where you might manipulate interest rates or assume you have control. You manipulate the interest rates and then you assume that you've controlled inflation. Education policy, maybe you raise test scores and that you assume you have improved learning, not necessarily. In mental health, you suppress a symptom and you assume you have resolved the condition. AI safety, you tune the behavior of the benchmarks and assume you have aligned the underlying model to what matters. The fallacy is not field specific. Again, I'm not attacking longevity research and I'm not attacking Sinclair. I just want to get the proper framing and the proper language around this, especially for laymen. It's a structural error in how we reason about high-dimensional systems from low-dimensional measurements. So naming the error can kind of matter. It gives you something. You might recognize that pattern. You give it a fallacy name. It's catchable, memorable. So how do you apply it? Well, when someone claims to have reversed X, ask, what are all the layers and which ones actually changed? Right? The general law is that perturbation is not a command, right? What do I mean? Well, in simple systems like a thermostat, a light switch, a gear, perturbation, where you change something, and command are kind of the same thing, right? You push the lever and then the system moves. And it moves in the direction that you intended, for more or less, right? So by the amount, in the direction you intended and by the amount you intended. So that's a good way to think of what a simple system is. In complex systems, an organism, an economy, an ecosystem, a brain, this equivalence completely breaks down. You can perturb the system, but you cannot command it like that. A command produces a predictable, intended, durable change in a system state. A perturbation produces a local response that is then processed by the rest of the system in ways you did not specify and cannot predict. Definitely not fully predict. Epigenetic reprogramming in retinal cells is a perturbation. It produces a local response. The epigenetic clock in that tissue moves. What the rest of the organism does with that perturbation over time at the systemic level, that is not yet known. The ability to perturb a complex system, yes, it's real, it's valuable, but mistaking it for the ability to command the system is where the distortion begins. Okay? Anyway, so I know I've gone on enough about this, but I'll tell you. We're not seeing age reversal. We're seeing the partial stabilization of one subsystem, measured through a proxy, under constrained conditions, mistaken for command of the whole. Okay? So, it's important that you pick up on that pattern. It happens all the time. We're going to see it again throughout Science in Perspective, and this is a good example of that. Anyways, go check out some of those links that I put in the description, and go check out dekyon.io, dekyon.io. I got a number of apps on there. One of them is dedicated to Science in Perspective. If you become a member for $9.99 a month, you get to learn a lot more about what I talk about. Data visualizations about the different concepts that I bring up and how they relate to each other. There's a study space if you want to dig a little bit deeper. Really for those who don't just want to be entertained, they want to actually learn and be part of a learning journey. There's a learning journey feature. There's techniques and mindsets. You can fold some of this stuff into your own life. All kinds of good stuff. Community forum, transcripts, other things that you might expect. Dekyon.io, go ahead and check that out. Okay, thanks everyone for listening, and for those of you who are watching, until the next episode, take care.